Hi, I am Axel Rose Rosal, and for today's video, I am going to discuss about glycoproteins. But before I am going to discuss about glycoproteins, allow me first to read the topic outline. So first, we are going to talk about the definition of glycoproteins, followed by the major classes of glycoproteins, and then later on the functions of glycoproteins as well as the difference between glycoproteins and proteoglycan. So first, let us proceed to the definition of glycoproteins. So what is glycoproteins? So glycoproteins are defined as a proteins that contain oligosaccharide chain or glycans that are covalently bonded. So basically, if we talk about glycoproteins from the word itself, glyco, it means sugar. So glycoproteins is a proteins with a sugar attached to it, and that is because of the presence of oligosaccharide chain or glycans, which is a sugar that is composed of 2 to 10 monosaccharides that are covalently bonded to a certain protein. Apart from that, we also have glycosylation process. Actually, glycosylation process occurs when the carbohydrates that is attached to a protein undergoes a certain modification. So actually, there are two types of modification. So we have the co-transitional modification and post-transitional modification. So if we say co-transitional modification, it is a type of protein modification that takes place during the translation or during the protein synthesis. So co-transitional modification mainly occur in the rough endoplasmic reticulum and is responsible for the regulation of translation, protein folding and processing, myristylation, and perennializations and palmitylation. On the other hand, if we talk about post-transitional modification, it occurs or it is a covalent or enzymatic modification of proteins that occurs after the translation or after the protein biosynthesis. So unlike co-transitional modification that only occurs in the rough endoplasmic reticulum, this post-transitional modification takes place in a several cell organelles. So it's such as in a Golgi body, endosomes, lysosomes, and secretory vesicles. Apart from that, it also focuses on the structural modification in a way that it happens via the additions of functional groups or proteins, proteolytic cleavage, or regulatory subunits, or by the degradation of the entire proteins. And then apart from that, characteristics of sugar may change the chemical characteristics of proteins that is required to function properly. So as what we all know, glycoproteins is a proteins with a sugar attached onto it. So basically, the sugar that is attached onto the glycoproteins has its own characteristics. So that characteristics may alter the chemical characteristic of the proteins to function effectively and efficiently. That is why oftentimes the addition of sugar is often required for a glycoproteins to function properly and reach its ultimate destination in cell or organism. And the next is glycoproteins are present or frequently present at the surface of cells where they function as a membrane proteins or as a part of extracellular matrix, in a way that these cell surface glycoproteins plays a critical role in cells, specifically in cell interactions and mechanisms of infections by bacteria and viruses. Now, let us proceed to the major classes of glycoproteins. So actually, the sugar that is attached to the proteins are located into two locations. So it's either in the endoplasmic reticulum or in the Golgi apparatus. Actually, there are two major classes of glycoproteins. So we have the N-linked glycosylation and O-linked glycosylation. But first, let us discuss the N-linked glycosylation. So if we say N-linked glycosylation, it occurs in the endoplasmic reticulum where N-linked sugars are produced, which we now refer to as an N-linked glycosylation. So as you can see in the presentation, I have here a picture wherein the blue one signifies an oligosaccharide sugar molecule, and the red one signifies as a nitrogen atom in the asparagine residue of a protein molecule. So basically, or in the other words, N-linked glycosylation is refers as the attachment of an oligosaccharide oligosaccharide sugar molecule, which is the blue one, to a nitrogen atom in the asparagine residue of a protein molecules, which is the red one. Now, apart from that, N-link glycosylation or this linking process is important in both the structure and function of some eukaryotic proteins, specifically in the archaea and rarely in bacteria, and that is because the added sugar residues are often used as a molecular flags or recognition signals to other cells that come in contact with them. And then apart from that, if we say N-link glycosylation, it undergoes a process of co-transitional. So it means that it happens during the translation. Moreover, there are important clinical application of N-link glycosylation. Like for example, it is associated with different diseases such as type 1 diabetes, Crohn's diseases, and cancers. Apart from that, N-link glycosylation is also involved in making the antibodies where it is a therapeutic proteins in the market. And then also, now let us discuss the O-link glycosylation. So if we talk about O-link glycosylation, this occurs in the Golgi apparatus where O-link sugars are produced, which we now refer to as O-link glycosylation. So as you can see in a presentation, I have here a pictures in which the blue one also signifies as a sugar, and then the red one signifies as oxygen atom of the serine or threonine residues in a protein molecules. So actually, N-link and O-link is actually the same, but they differ where the sugar is being attached. So O-link glycosylation is also referred as the attachment of sugar molecule, which is the blue one, to the oxygen atom of serine or threonine residues in a protein molecule, which is the red one. So apart from that, O-link glycosylation also undergo a process of post-transitional modification that happens after the synthesis of the proteins. Apart from that, this also occurs in the both eukaryotic and prokaryotic. Moreover, during the O-link glycosylation, several sugars can be added to the serine or threonine, in which that addition may affect the protein in a different way. So it's either in the changing of the protein stability or by regulating the protein activity. Now let us proceed to the functions of glycoproteins. So actually, glycoproteins are involved in nearly every process in cells, so which means that they have a diverse function such as in our immune system, protection of our body, communication between cells, and our reproductive system. Now let us first discuss the immunology, or how does glycoproteins affects our immune system. So glycoproteins has an important role for white cells, because if we say white cells, it rolls along our blood vessels looking for a potential invaders, but the way they attach to the blood vessels lining is through the glycoproteins called lectins. So basically, if you're a person who has lacked lectins, as a result, or it is expected that your immune system would be pretty weak, since your white blood cells wouldn't be able to travel the body. Apart from that, glycoproteins are also important for red blood cells, specifically the blood type, because the blood type are the one who will determines the type of glycoprotein that is present in our red blood cells. That is why if you're a person who has a type A blood, you have a antigens or A glycoproteins on your red blood cells, in which this helps the body to identify that your blood is part of you and tells it not to attack you. Moreover, glycoproteins also helps to stimulate the process of coagulation of platelets to clot the bloody when you got cut. So that is why people who are missing important proteins on platelets can clot their blood and have a disease called hemophilia, where any cut continues to bleed independently. Now let us proceed to the protection, or how does glycoproteins protects our body. So first is the mucus. So as what we all know, many organs in our body need to secrete mucus to function efficiently and effectively, and this includes the stomach, the small intestine, and the airways in the lungs. So cells lining this body cavity secrete or send out glycoproteins. So the sugar mixed with water in our body create a smooth mucus. So that is why in the stomach, this mucus helps protect our stomach lining from the harsh acids needed to digest food, and then in contrary to that, in the lungs, the mucus help to trap bacteria, keeping our lungs clean and healthy. Moreover, glycoproteins are also involved in keeping our skin healthy, specifically the epithelial cells and the cadherens. So, epithelial cells is a type of glycoproteins that helps in a way that it attach our skin cells to each other, forming a tough barrier to protect our body. And then on the other hand, if we say cadherens, it is a glycoprotein that helps our skin hold together. So actually, I have here a presentation or a illustration of cadherens. So as you can see in a presentation, the long black lines connecting two skin cells together are types of cadherens or kanang naay arrow. So cadherens, as you can see in the presentation, cadherens are the one who holds the skin together. And then moreover, let us discuss the hormone. So actually, there are many glycoproteins that functions as a hormones, such as the human chorionic gonadotropin or HCG, which is present in a human pregnancy urine. So this HCG works in a way that it stimulates the corpus luteum to produce progesterone to maintain the pregnancy. So basically, this has something to do with the hormones of the pregnant woman or hormones of the girl. So apart from that, another example or another function or another glycoproteins that acts as a hormones is what we call as the follicle stimulating hormone or the FSH, which is also a gonadotropin hormone that has a several function and development, growth, puberty, and reproduction. Apart from that, another example is the erythropoietin. So it is a cytokine secreted by the kidneys that stimulate red blood cells productions in bone marrow in high levels in response to hypoxia or low levels normally. And then also glycoproteins also has an important role in a reproduction because through glycoproteins it increases a sperm cell's attraction for the egg by altering the electrophoretic mobility of the plasma membrane, or in the other words, because of the glycoproteins, it allows for the binding of the sperm cell to the surface of the egg cell. So that is why that there are what we call as offspring. And then apart from that, zona pellucida is also mentioned. Actually, zona pellucida is also a glycoproteins that surrounds the egg and prevents polyspermy from occurring after the first sperm cell has penetrated the egg's plasma membrane. Moreover, in reproduction, glycoproteins called hen ovalbumin are also mentioned. So actually, hen ovalbumin is a glycoprotein that is found in an egg white that serves as a food storage unit for the embryo. So now let us discuss the difference between glycoproteins and proteoglycans, because oftentimes people use these two terms as if they are the same, but actually these two terms are actually a part of, or they are different. So now, as you can see in the presentation, you can clearly, or you can vividly see that glycoproteins and proteoglycans is really different when it comes to the structure. So now, let us define, or let us differentiate these two terms when it comes to the definition, the structures, the carbohydrate content, the carbohydrate chains, as well as the location and the rule. So first, let us discuss the glycoprotein. So when it comes to the definition of glycoproteins, a glycoproteins is a proteins which is with a sugar attached on a structure, whereas if we say the proteoglycans, It is a protein molecules containing many bound glycosaminoglycan chains. And then when it comes to the structure of these two, glycoproteins have a short, highly branched glycan chains with no repeating units, whereas proteoglycan have a long unbranched chains with disaccharide unit as a repeating structures. And then apart from that, the carbohydrate content of glycoproteins is about 50 to 60%, whereas the proteoglycan is about 10 to 15%. Also, the carbohydrate chains of the glycoproteins may or may not be negatively charged, whereas the proteoglycan's carbohydrate chains are negatively charged due to the presence of the sulfate and uronic acid groups. And then the location glycoproteins, as you can see in the picture, it is located in the cellular membranes of the cells, whereas the proteoglycan, as you can see in the pictures, it is mainly found in the connective tissues. And then the role of glycoproteins is to help in cellular recognition, whereas the role of proteoglycans is important in the modulation of cellular development processes. So that ends my report, and thank you.